Okay. Nervous system continued. Uh, Parts of the cortex, you need to know frontal, parietal, occipital, and temporal lobe. Each of them control certain things. I would know what they control. There's two areas, Broca and Wernicke's area. Broca's area is where speech is generated, and Wernicke's is where it is heard. And so sometimes when people have damage in those areas, it either impacts their ability to form like sentences or to understand language. Um, the cerebral cortex receives input from all the sensory organs and basically captures that and processes all of it. Then the thalamus directs that input to specific locations. Um, when we look at the cortex, specifically when we look at the somatosensory, so the two strips in the middle of our brain, it's gathering all of this sensory information and then processing it for us. This is just showing like where on the cortex we get sensory information from, right? Um, so the different lobe functions, frontal lobes are like emotion and judgment, and we call them executive functions for making decisions. This is why when people drink alcohol and their decision making is poor, the frontal lobe is what is being impacted. Um, so now we've focused in on the nervous system. How does this react to like the bigger picture? We know that muscle contraction is a type of response generated by the nervous system, and they're interdependent. When I look at a muscle, I can break it down into parts. My full muscle, so like a bicep, the bundle of muscle fibers. A single fiber is the cell. They are like long, thin cells with multiple nuclei. And we go organ, tissue, and the cell is the single fiber. Within that single fiber, we have basically two light bands and a dark band. All of these units are called sarcomeres. So we look at this, right? Basically, what it is it's these myosin and actin filaments layered on top of each other, and they will join together and scrunch and make the muscle shorter. This is why, if we remember, the A band or the dark band gets like shorter and the I bands, the light bands go away because everything is squishing together. Do this if you want to. This is the correct answer. Right, we're looking at relaxed A versus contracted B, and we see that basically all of the light bands have gone away because the myosin and actin are pulling towards each other. This is again another example of that. Muscle contracted. Now we see everything's overlapped. We can see the big difference. And so that is what is happening during contractions. What is happening at a molecular level? So if we look at one myosin, thicker, and one actin, the thinner, we see that we have a myosin head. What happens is ATP is going to attach to that myosin head, and the myosin head is going to move and attach to the actin once I've put in ATP, which is the energy. The myosin head is going to create what we call a cross bridge. It's going to attach to the actin, and then it's going to contract basically downwards or up and away from the myosin, pulling the thin filament in a certain direction, which is causing the contraction. Once I've used that ATP, it goes away, and I need more ATP to make it happen again. This is just a zoomed in version of that. Again, the P is just showing, right, what we now know is that when I cut off a phosphate, I'm breaking that bond, I'm releasing that energy. And it looks like something like this, which is kind of a cool image, right? The green is going to be my phosphate, that is going to be my myosin head, and the actin filament is going to be the yellowish orange. To prevent contractions from happening at certain points, calcium blocks the myosin binding sites on actin. And so muscles cannot contract when calcium is within those cells. The arrival of the action potential is going to draw the calcium out of the cells, allowing the contraction to happen. And this is just looking at the relationship between the neurons and the other parts, the muscle. Okay, last one. Okay, specialized receptors. We now hopefully know that these 4 lobes, temporal is going to be auditory, occipital is going to be vision, parietal is all of the processing, and frontal is decision making. So we're focusing on 2 of them. So for occipital lobe, the human eye is really fascinating, right? Because we know that it actually inverts what we are seeing. This is the anatomy of it. What we know is that at the very back, so not right where the optic nerve is, but all along the back, I'm pointing at it thinking you can see this, all along the back of the eye are what we call rods and cones, which are going to be which are the cells that receive the image and determine like color and things like that. If we look, the light, the little cones at the very bottom of this image is the visual going in. Basically, the rods and cones are going to receive that message, and then they're going to send them to the like the ganglion cells and stuff like that. So the green, the brown, the orange, the purple, because that's going to go to the optic nerve. So it basically goes all the way to the back of the eye and then comes back out and is sent in a message to the optic nerve where all of that visual image is concentrated and sent to the brain. Again, it looks like this, right? So this is showing us that the light shines in, my rod and cone receptors activate, they light up, it sends the message from the ganglion to the optic nerve and to the brain. This is rhodopsin. If you remember when we went on our senior trip, they talked about rhodopsin, which basically allows us to see at night. This is just an example of how our eyes are capable of adapting to seeing at night. Rhodopsin is a chemical, basically a neurotransmitter or a hormone that's released that adapts our eyes to seeing at night, but when we see a light, it decomposes and we cannot see anymore. Retinol is another chemical that is necessary for vision as well. We won't really get into it. So moving on to the temporal lobe, but blind spot right where the optic nerve exits, there's no rods and cones. Oh, okay, so I guess we're not moving on to the ears yet. So the lens is basically allowing us to see things. Depending on how it is tilted and stuff will determine what we see. Okay, human waves. Takes sound waves, translates them into a mechanical signal, and then to an electrical signal. These are the parts: outer, middle, and inner ear. The pinna is basically your physical ear. The auditory canal is like what you stick your finger in. And then in the middle and inner ear are these like little tiny parts. We have our eardrum, and that's hooked up to like the stapes and the hammer. That's number 6, all of those tiny bones. That is hooked up to the cochlea, and the cochlea is what turns the mechanical movement, the vibration of the eardrum, into a electrical signal that goes to your auditory nerves, and then eventually to your temporal lobes. And that is it for the nervous system. We'll talk briefly about the immune and the endocrine system next.